forgetting to mention, and I know a lot of you have already noticed because I've gotten responses, but I also put up a survey with every homework question, with every homework set. It's not required, it's just to help me. Like if there was a question on the homework that you really didn't understand and you essentially guessed to get the answer, let me know by that, and then I can spend more time in class that day on that topic. All right. So we essentially will finish up with what we had from last time. So we'll kind of finish the microbiology history lesson, and what we're gonna get to now is essentially where do microbes fit in like the organizational scheme, right? So we had talked about discovery of microbes and how they played a role in different diseases and things like that. But now now that scientists had found these things, they need to figure out how to classify them, how to categorize them. It turns out it's a bit of a challenge to classify them because essentially they kind of look the same, but they do very different things. So it was kind of hard to do that. And it was also kind of hard to track the DNA because you can just essentially, they can pass DNA along to each other. So it was quite difficult for them in the beginning to figure out how to actually classify microbes. So some of the ways that they came up with were using biochemical properties of them, right? So things like the gram stain, which we'll learn shortly about in labs, and then what they can actually do. So their ability to metabolize different substrates, those are also different ways to categorize bacteria. In the end, one of the most useful ways is going by their DNA sequences. All right, so kind of going, again, this now taking a look back through history, right? So again, in the beginning, they just had plants and animals. And then now they discovered all of these microbes and they didn't know what to do with them. So they defined a third kind of life and called it this Monera group, right? But then eventually they started figuring out that there was more to it than that, right? That there were actually eukaryotic type microbes, right? So the protozoa and the algae. And then there were the prokaryotic types. So then it split again. So now every time they're learning more something about it, the definitions get more specialized and it starts splitting them up into more groups. And then finally they decided that fungi would be its own other kingdom, right? And so you've probably heard of the classifications before where they're called the 5 different kingdoms. And so that is kind of what held for quite a while. And then came along Lynn Margulis. So what she did was she kind of turned everything on its head a little bit and had this idea that actually eukaryotes had evolved from prokaryotes, right? So this idea that they essentially merged with bacteria cells, right? So this idea of endosymbiosis, right? So that one cell essentially internalizes another, and then that's how it evolved to have this other one. So the main ones that she had proposed, again, were that mitochondria had evolved from bacteria and that chloroplasts had evolved from cyanobacteria. And so, and then, there was a lot of evidence that actually, people have actually been able to show a lot of evidence to prove that this was right. But what we've kind of finalized now on was essentially set by Carl Woese. So he was studying archaea and operating kind of under that 5 kingdom classification with the ideas that Lynn Margulis had proposed. But he was studying these archaea, and it was really hard for him to classify it under what had already existed, right? They kind of looked like prokaryotes, but then they had some functions like ribosomes and things that were more similar to the eukaryotic ones, and then they kind of lived in these crazy places, and he was like, well, I can't really fit this into what the existing definition was. And so also what was happening around that time is we had talked about DNA sequencing, right? So when Sanger developed sequencing, now you have this ability to essentially look at the genomes of the different microbes that you're studying. And Carl Woese decided that looking at this 16S ribosomal rRNA gene was actually a good way to compare the different species. And so he used that to come up with these the three groupings that you have here, which we know of now, right? So the bacteria, the archaea, and the eukaryotes, right? But one thing to note is that, so even though he had split these, and all of these relationships you can go back to and trace based on this 16S gene, but you'll notice that it actually still keeps the same ideas that Lynn Margulis had about the proteobacteria and the cyanobacteria essentially evolving into the different eukaryotes. Okay? So any questions on this? So this is like wrapping up from last lecture. Kind of finishes our history lesson. Okay. So we'll move on to the goals for today's lecture. Today we're going to essentially jump into the structure of bacteria. So there's different components, and we'll specifically go into today the cell envelope and how do you compare like gram-negative and gram-positive bacteria based on the components that essentially make the cell itself. I also posted this background reading section. It might be a little bit confusing because it has some stuff about like eukaryotic cells that you don't really know, but there are things that you'll notice when we get to like lipid compositions and kind of like talking about transporting molecules across the membranes that this other resource is good for. All right. So we're going to talk about, again, cell structure today. So what does cell structure mean? Essentially, all of the components, like, so what the cell is made up of, okay? In this class, a lot of times what we're going to use is E. coli as the model organism, because essentially we know the most about E. coli, and it is a very good model organism, especially for gram-negative bacteria. When we start talking later on, we'll pull in some more examples from gram-positive bacteria, but essentially, a lot of the topics that we're talking about today will be based on what we know from E. coli. All right, so today we're essentially going to talk about the different structures and the different components of this cell that we see, and I'll go through them individually. All right, so in the beginning, a bunch of definitions. Hopefully you've seen a lot of these in 106, 107, and so it shouldn't be new. So the cytoplasm essentially is what's inside the cell, right? So is where essentially all of the stuff happens in the cell is inside the cytoplasm. It contains a bunch of proteins and other macromolecules. In here, you'll notice that's essentially where all the ribosomes are. So that's where all the gene expression is happening. Then we have the cell membrane. So the cell membrane is essentially what encloses the cytoplasm. So that's essentially kind of starting to define the shape of the cell. And then the cell wall goes outside of the cell membrane, and we'll get to more pictures where they're more clearly defined. But so again, the cell wall provides more protection. Again, usually that's what the main function of the cell wall is, to provide more protection for the cell. And so that goes outside of the cell membrane. All right, the nucleoid. So this is the essentially regions inside the cytoplasm that contains the chromosome of the bacteria species, right? So all the DNA essentially it's coiled up inside the cell, and that's where the nucleoid is. It's not in this picture, but we will talk about this a little bit probably tomorrow. Monday is the flagellum. So certain bacteria have the ability to move via these flagellum. Essentially, they look like tails coming out of the bacteria. They rotate and allow for movement. All right. So the point of this table is essentially to show that the major components of the cell, right? This is percentage by weight of the cell, right? So essentially the cell is full of water, right? That's the major component. So inside the cytoplasm, you can think of it as a fluid, right? So it's water. And then there are a bunch of these other components, right? Proteins, RNAs, the different lipids, DNA. And then there are also the essential ions, right? And the organic molecules and things like that that are inside the cells. In general, all different species kind of have these category of things. But depending on the species, and even for one single species, depending on how fast it's growing and what kind of stage it is in its growth cycle, the ratio of these things is going to change. So this is just, one snapshot of E. coli during exponential growth. Okay, and so this is just to give you a feel for it. Most often, right, water is still going to be the biggest component, but all these other things, how the ratio of those might change depending on the growth cycle of that species. All right, so the first thing we're going to talk about is the cell membrane. So again, the cell membrane essentially is the thing that defines the existence of the cell, right? So, most of it, again, the inside is mostly water and things like molecules that are kind of floating around in there. So it needs something to encapsulate it to define the actual cell itself. So that's what the cell membrane does. So in this picture, and we're going to go through all the details again and again, but you'll see that it's made up of this lipid bilayer, right, that has all these different proteins kind of in them, and each protein has a different function, and we'll get into what some of those functions are. But so we'll first start with what the actual cell membrane functions are, right? So again, it's a physical, mechanical boundary, right, between the cytoplasm and the outside world, right? So the cytoplasm's on the inside, and then you have your extracellular environment. So it is selectively permeable, meaning that, and this is for its own protection too, right, that things just can't go through this, right? So if you have something on the outside, it's not everything can just pass through the lipid bilayer, right? And that, again, it serves as a first line of defense for the bacteria, right? But as you can imagine, it needs to get stuff from the environment, and so we'll start talking about how it actually gets the things that it needs. So again, starting to talk about that. So protein transport, so nutrients and waste. So that's what some of these, the proteins that are in the bilayer are for, so that it can actually allow the things that it wants in, and it can actually kick out things that are bad for the cell as well. Things that we'll talk about later on in the class, it's the site for electron transport change, so how we do respiration and photosynthesis. Again, it's also involved in lipid synthesis itself, so that the lipids that's involved in here. And then the other thing that it can do is, so these proteins, some of these proteins are actually used for the bacteria to actually sense its environment. So it can detect what's outside, and so that will essentially determine what the cell does, whether it moves, whether it makes this other thing, whether it tries to let something in. All of that is done essentially based on the things that are embedded in the cell membrane. All right, so the first component of the membrane that we're going to talk about are the membrane lipids. All right, so this is just a toy structure of what the lipids look like. So everything has this phosphoryl head group. So this head group here. So this is it's charged, right? So this is the kind of hydrophilic or the water-loving end of the molecule. And then you have essentially the fatty acid side chains down here, so these things here. So this is the nonpolar end of the molecule, right? So these are the things that are hydrophobic, okay? The other key thing here is that, so what links the phosphate group to the actual fatty acid side chains is this glycerol derivative. And it's actually linked here by ester bonds, and we'll keep talking about that, why that's important, and a little bit more details on that soon. But any questions on the general structure of this? All right. So again, as I mentioned, what links the fatty acid group to the glycerol is a ester bond. And the key to this is that you'll notice that it's in bacteria and eukarya. This is important to remember because when we get to studying archaea, this bond is actually not an ester bond, it's an ether bond, and that's how you can actually tell them apart. It's one of the distinctive features of them. Okay, so again, this one is just showing one of the side chains, like what the chemistry looks like for one of the side chains, but essentially here you have your ester linkage linking it to this glycerol derivative. All right, so essentially based on the structure of an individual phospholipid, because of the fact that you have the hydrophobic ends and the hydrophilic ends, it self-assembles into this, what we call as a lipid bilayer, right? Because essentially, remember, the cell is mostly water, right? So if you were to just have these single species like just floating around, it's just not gonna happen. It's not gonna like that, right? Because there's water everywhere. And so what it does is it forms this bilayer. So now the two non-polar ends that don't like water are facing each other and they're protecting itself essentially from the water. And then you have the hydrophilic ends, and so there's aqueous environment on the inside of the cell, and usually there's aqueous environment on the outside of the cell, and so these ends are also happy, so they naturally will form this lipid bilayer. And then just more terminology, right? So the different components, so the one that's more towards the extracellular environment is called the outer leaflet, and then the one that's closer to the cytoplasm side is the inner leaflet. All right, so now just to kind of compare two different phospholipids, right? So there are many, many different, you can have many varieties of these phospholipids, okay? So the general things to note, right? The phosphorohide group, right? So the phosphate group here. But as you notice in these two, they can actually be, what's on the top can actually be a different group, okay? Again, so the common thing is this glycerol derivative with the ester linkage down to the actual fatty acids, but these fatty acid side chains can be different in this case. In these cases, the only difference between these two is the actual length, so how many carbons are in the actual side chains. But again, so even in a single cell, you might find several different types of these fatty acid side chains or these phospholipid groups. All right, so another way that these different phospholipids can vary, so we talked about length, right? So the length of the chain is one way for it to vary. The other way is if they have here, so in this case, this first one is double bonds, right? So whether or not they are unsaturated. So most of these double bonds, so in this case, you'll see that they're in trans like this. So trans meaning they're on the opposite ends, right? Of the carbons are coming out from the opposite ends. In some cases, you will have these cis double bonds, so it's on the same side. So what this actually does is creates this kink here. So in both of these cases, these are both double bonds, unsaturated. This one causes these kinks. And then you can also have, sometimes you can have these cyclized carbon rings in them. Again, so each different cell will have the ability to make these different kinds of phospholipids based on how they evolved and what environment they're living in, and they can change what their cell membrane looks like depending on conditions. All right, so one of the ways that scientists describe the actual cell membrane is this note of this fluid mosaic model. So they decided that, this was just a term that they used to describe what the cell membrane was. Essentially, it was this mosaic of components, right? So it had the lipids, it had all these different proteins in it, right? But they were essentially fluid because they would kind of move, the components would move around, right? And that movement is actually necessary for the cell to function properly, okay? And so the kind of tie to what we're talking about now is, so how do these actually get membrane fluidity based on the different components inside the cell membrane? So the key to that is essentially the makeup of the different phospholipids in the bilayer. All right, so we're going to talk about two things. One, in ways in which you can increase membrane fluidity, okay, so making things more movable. And so how you do that is essentially shortening up the chain length, so using shorter chain lengths in how many carbons there are, and then more double bonds, so the more unsaturation. So essentially, if you can think about, so why would double bonds increase fluidity? Essentially, they're making gaps, right? And so if all of the lipids are trying to like pack really tightly together, now you throw in a kink in there, it's making some space. There's more space for the proteins to move around in. The membrane is more fluid. Okay, so now on the other hand, we want a way, the cells need a way to actually make the membrane stiffer, right? So reducing fluidity. So again, so it's the opposite, right? So longer chain lengths. So this actually essentially increases the van der Waals interactions between the lengths of the carbons, right? So those interactions are gonna kind of stiffen up the membrane. Again, fewer double bonds, right? So making everything straight in there, packing everything in tightly. And then you can add some of these different molecules in there, right? So the stiff planar rings, so the cyclized rings in there in the lipids will actually make that stiffer. But they also have these added components. So in eukaryotic cells, we have the sterols. So the most common one that you hear of in the membranes is cholesterol. The essentially prokaryotic equivalent are these hoponoids. So these things here, essentially these molecules go in here, and again, the way you can think about it is they're essentially locking up the membrane by making it stiffer and not allowing things to move. Yeah.
What's the reason behind doing the membrane to decrease the membrane or increase it? Like, what's the reason, if, let's say, what we're trying to achieve by decreasing the membrane fluidity or increasing it?
Sure. So the first thing is, well, depending on the environment that the bacteria will see, right? So the easiest one, and this kind of has to do with the question that we're going to go through next, is the temperature of the environment that it's living in, right? So what do you think you would want the cells to do if it all of a sudden found itself in an increased temperature environment? Would you want the cells to increase or decrease fluidity at that point? Why do you say increase? So what's happening at high temperatures? Destabilizing. The things are essentially, so if you think about like just melting, right? Like things are starting to melt, right? So if things are melting, do you associate melting with something that's more fluid or less fluid? Something that's melted, if melted, is that more fluid or less fluid? More fluid, right? So if you want to counteract that, you want to make it more stiff, right? Okay? Somebody else raise their hand. No? Okay. All right, so does that make sense? All right. So that kind of gave away half of this question, but so I want you to think about, how would the bacteria adapt to increasing temperature based on this, right? So think about your answer for a second. Talk to a neighbor. I want you to share with them your answer. Even if you have the same answer, I want you to explain to each other why you have that answer, because you might have a different reason. Okay, and then so talk about it for a minute or two, and then we'll go through the answers.
Because basically what you're saying, when T is at higher temperature, this cell is one of these so it has to like have access to that out of like that high temperature is very easy to do. They would end up like
All right, so what do you think? So the first question you have to ask, so when you increase the temperature again, so what does the bacteria want to do? Okay, so it wants to be more stiff, right? It wants to decrease the fluidity in its membrane, so how is it going to do that? So what's the answers? E. Yep, so by both A, so increasing the longer chain length and decreasing the number of double bonds. Is that clear to everybody? Yeah. Oh, sorry, hold on. Go ahead.
Can you use cholesterol as well? Can you use cholesterol as well?
Yes. Yep. So it just wasn't one of the options, but yes, absolutely. There was a question from over here. Not anymore. How about up here?
Sorry, did you say it was A?
So it's E. So it's both A and B.
But doesn't it use long chains to become more fluid?
To become more stiff. Long chains are to become more stiff. So the longer the chain, the more van der Waal interactions, and so they're kind of locking in there. Okay? Okay, make sure you understand this. This is an important concept. Yep.
So the increase in temperature, is it like a spontaneous chemical reaction thing that happens, or is it like actually something that's something that the cell itself kind of sends out proteins or something that make it happen, like breaking bonds and stuff?
So the case that we would think of, right? So all of a sudden, for whatever reason, the bacteria is now in a warmer environment. Like, and the temperature here, it changes like 30 degrees in one day, right? So all of a sudden it was cold and now it's hot. Okay, so it's going to sense that the temperature has increased, and it's going to say like, oh, well, maybe some things are starting to melt and that's not good for me. And so it's going to sense that, and it's going to start triggering production of the different kinds of lipids for the membrane. And if you think about it, and we'll get to this in a couple of days, but we'll start talking about how fast bacteria actually grow and divide, and that's happening, for E. coli, that happens in 30 minutes, right? And so even if it's not changing the composition for itself, when it starts dividing, all of that process is happening at the same time, and so the ones that are growing are going to be able to survive better than the one that is. All right. Okay, so more about the membrane proteins. So again, we kind of already talked a little bit about the different functions that they have, but again, they also provide structural support. So just the fact that it's like locked in there, it's also providing some kind of rigidity structural support to that. One of the major things that I had already talked about, it detects signals from the environment, so it can sense what's happening in the changes outside. So in this case of temperature, there's likely something out there that's being able to detect that. Secretion of virulence factors and communication signals. So again, the proteins that are in these membranes are in charge of things coming in and things going out. And so whether that's a signal to some other cells, or it's just getting rid of waste from the cells, that's what they're there for. So that's a similar thing for ion transport. And also for energy storage, again, that's what we'll talk about later in the class when we talk about metabolism. And just to give you a feel, right, so proteins take up about half of the membrane, the mass of the membrane. So there is a lot of proteins in the cell membrane. All right, so again, one of the things we talked about, one of the purposes of the membrane is to be this barrier, right? It's a physical barrier, and a lot of times that's for its protection. But the cell needs a lot of different molecules, nutrients, ions, and things like that. And And so a lot of that can't actually make it through the membrane. So there are different classes here. So certain molecules that are able to actually freely permeate through the membrane are these hydrophobic molecules like oxygen, carbon dioxide, and nitrogen. So those things can just kind of pass through. Small uncharged, so polar molecules like water and glycerol, so those can get through, but it's pretty slow, and some of it will make it through. In most cases, not enough for what the cell needs, and so the cell has actual proteins that it'll use to get these things across. The larger molecules definitely not getting through, right? So it just can't make it through the membrane, and so it definitely needs a way to get in. The same thing goes for ions. These charged molecules, they're not actually able to make it through. So again, that's why it's called this selectively permeable barrier. So again, now it can't make it through on its own, but the cell needs it, and so the cell has, again, these proteins that are essentially these transporters that are able to pass material through the cell. Sometimes it can be through passive transport, so where things just kind of flow through based on the concentration gradient, so whether the outside or versus what's on the inside has a difference in concentration. So it'll always go from high to low, and that's passive transport. In the case of active transport, even if there is, more of something inside the cell versus outside, the cell still needs it for some reason, and so then it has to go against the concentration gradient, and it'll actually require energy to do that. So it requires some form of energy, and we'll get into those kinds of specifics in the upcoming chapters. All right. Okay. So now I just wanted to step back and kind of figure out what we've been talking about. Okay. So this is a picture of the E. coli cell, right? So this in here is the cytoplasm, and then we have been talking essentially about this membrane here. Okay. So this right here is what we call the cell envelope. So this kind of zoomed in section here is this of E. coli. Is the cell envelope, and so we've only been talking about this one component over here. So we're going to start now talking about the rest of the components on that are outside of the cell. So the first thing that comes through, the next kind of component is the cell wall, and so the cell wall is all right. And so the first thing that's found in most bacteria, it's it's essentially what gives it more protection and structure. So it helps withstand different Turgor pressures. So essentially, it kind of helps the bacteria not explode. But essentially, so another name for the cell wall is the sacculus, and it's made up of a single molecule called peptidoglycan. So again, so cell membrane, and then on the outside of that is the cell wall, and so that's what we're going to talk about now. All right, so this is what the cell wall structure looks like. So this is a peptidoglycan structure. So peptidoglycan is essentially made up of these two different sugar disaccharide molecules. So it's the N-acetylglucosamine and the N-acetylmuramic acid. So the purple and the blue. Right, so it's essentially a repeating structure of this like purple, blue, okay? And that's when we make this long chain, and that'll go all the way around the cell. You'll notice in here, so that now these are actually cross-linked. So these are cross-linked by 4 amino acids. So here's the 4 different amino acids. And the unique thing about the prokaryotes and their cell walls is that they contain these D forms of alanine and glutamic acid. Okay, so your normal form that you see in proteins is the L form, right? And so these actually contain the D form, and these form these different peptide bridges. All right, so it's this other amino acid, this diaminopimelic acid that actually is able to create this cross bridge here. So in the picture before, you can see they kind of have them like overlapping. So this other amino acid is what's actually allows it to make this cross bridge, and so it links the different chains together. So you have, the alternating sugars going here, and then you have it actually linked together, and that kind of creates a little bit more firmness and rigidity in the wall. So it's this linking here, the linking of the peptides. You have the 4 different amino acids that are coming out of the ends, and then they will actually link together and form this cross bridge. So these pictures right here. And so the fact that the cell wall is made up of and has these D components, right? These D amino acid components. This is what actually makes a good target for antibiotics, because our cell walls don't have, we don't have these D amino acids, and so you can have these antibiotics that will specifically target essentially the synthesis of the cell wall here, and so things like penicillin and vancomycin. So because of the fact that the bacteria are using the D-alanine, we are able to have these different antibiotics that can target that synthesis, and so they can target the bacteria only without harming us. Any questions about this? Oh.
Couldn't certain antibiotics have an effect on the bacteria and the good that we have?
Yep. Yeah, so something like this, right? So something like penicillin, vancomycin, it's going to attack every single bacteria that has a cell wall, right? So it's going to attack every single one of them. They're not specific. So yep, it might be attacking the bad ones, but it also might be attacking the good ones that are helping us out. All right, so now we're going to start talking about the whole picture, the big picture of the whole cell envelope, right? So there are 3 major kinds of cell envelopes: the gram-positives that have thick cell walls, the gram-negatives have thin cell walls, but then they also have other components, which we'll talk about, and then the mycobacteria are ones that are essentially, they're really complex. They have all these different components and different structures that the gram-positives and gram-negatives don't necessarily have. All right, so the first thing we're going to do is we're going to talk about the gram-positive cell envelope. So you'll notice, so we already talked about the cell membrane here, right? So this is the cell membrane. So we talked about the cell wall, so this is the cell wall. But going from outside in, so this is the extracellular space is essentially out here. We have this S layer, and so not every single species is going to have this, right? So in this case, we're showing that this gram-positive bacteria has this S layer. So it's again made up of different proteins that goes on the outside of the cell wall, and it has these different pores, so it can let things through. But the S layer, again, just remember context where that is. So again, then we have the thick cell wall, right? So the gram-positives, you can imagine that they have more layers. So it's like these 3 to 20 layers of this peptidoglycan. So one of these here is essentially a layer. So each row like this is a layer, right? But the other thing is that they also have these blue lines, essentially, these blue lines here that are represented here are these teichoic acids. And so that again adds more strength. So this is what it actually looks like. I don't expect you to remember what the chemical structure of the teichoic acids is, but just to know what they're for, right? So they're in there in between in the peptidoglycan layer, and they're providing even more strength and rigidity to that cell wall layer. All right, so for the gram-negative cell envelope, so remember when we had the big picture for E. coli, we were talking about the cell membrane. There's like there's all these different layers right on the outside of the gram-negative layer. So the first big difference is that it has this outer membrane. So it has this second membrane here. So this is the outer membrane here. This actually goes on the outside of the peptidoglycan layer. And we'll go through more details about what this outer membrane is in a second. But in the case of gram-negative, so they do have this cell wall, so they do have a peptidoglycan layer, but it's very thin. It's just maybe 1 layer, 2 layers. Okay, so it's nothing in compared to what the gram-positives have. But now that you have essentially this outer membrane, right? So now you have more space in between. There's another definition of space in between. So inside here again is the cytoplasm, but now the space between the inner membrane and the outer membrane is this periplasm. All right, so now more specifics about the outer membrane. Okay, so the outer membrane, again, so you can think of it similar to the cell membrane in that it has the outward facing leaflet and the inward facing leaflet. So in this outward facing leaflet, it has what are called these lipopolysaccharides. So these are endotoxins. So this is one form of defense for gram-negative bacteria, right? So these endotoxins here can actually cause us harm. And so it is a form of defense that they have. This lipid A, so this lipid A here, so you'll notice, okay, some of the similarities. So in here, it looks like, oh, this looks just like the cell membrane, right? It has these phospholipids here with the similar structure that looks like, so that's true. So there are different phospholipids in this outer membrane, but what it also has is this lipid A that you see here that has all these 6 fatty acid legs. So there's something slightly different, and that's what actually connects to this LPS molecules here. Okay, and then from now, so on the inward facing leaflet, so this side here, we have the lipoproteins. So these are the ones that are now connecting the outer membrane to the inner membrane. So now when we talk about gram-negative bacteria, you now call it the inner membrane, so that's the same thing as cell membrane. So it's just to be able to make sure that you differentiate from this outer membrane. So if you ever hear inner membrane for gram-negative bacteria, just note that that's the same thing as the cell membrane. Okay. The other thing that you'll notice, so now, okay, we had already talked about there are some proteins inside the cell membrane to get stuff in, right? Now there's an even bigger problem because now there's an extra membrane that things have to go through. And so again, there are more proteins also in the outer membrane that'll allow different molecules in. The names for the ones in the outer membrane are called porins. So again, they allow stuff in like sugars and food, energy, things that the cell needs, but they also are a way in for antibiotics as well, for certain classes of antibiotics. All right, so the key is that, and this was one of the things from the homework that I think confused people, was that this outer membrane is gram-negative only. Okay, so this is the only exists in gram-negative bacteria. Okay. All right, any other questions? That's essentially, well, so you'll learn this in lab, but there's a thing called the gram stain, right? And so the gram negatives are negative for the stain and the gram positives are positive for the stain, but it's based on the structure of the cell envelope. For today. Yep.
What exactly does gram-negative